Our next speaker is Briony Yorke, who has recently and happily, she emphasizes, moved to Leeds as group leader. She's going to tell us about UV and X-ray crystallography.
Okay, hi everyone. Thank you to Helen for inviting me to talk, and also, as you'll see, for quite a lot of support in my research using the tools that she's created to look at changes that we see in time-resolved crystallography. So first of all, this is one of my favorite quotes from the history of protein crystallography. Crystals don't wiggle, and if it doesn't wiggle, it's not biology. But as we all know, crystals do wiggle, and proteins in crystals wiggle quite a lot. That's why we have thermal B factors. That's why sometimes we have missing parts of our structures. Proteins in crystals are quite dynamic. And thankfully for me, that means we can use that to look at how they change over time in response to perturbations. And so traditionally, the idea of protein crystallography has been that it can help us to get a relationship, to understand the relationship between three-dimensional structure and biological function. But what time-resolved crystallography does now is it helps us to bring in this third thing. So how does the structure determine dynamics, and how do they both together inform function in biology? And dynamics is actually very important. So when we do time-resolved crystallography, we tend to do an experiment that's based on the experimental design that was first introduced for flash photolysis in the 1940s. So this is called the pump probe experiment. We perturbate our structure at time 0 using a trigger, often light, and after a particular time delay, we probe that using X-rays. For crystallography, we then have to repeat at multiple orientations of the crystal to sample reciprocal space and get our structure. And then we repeat at different time delays to see a series of changes that occur over time. But there are limitations to this method, important limitations, important considerations that we have to think about when we do the experiment. So first of all, Elspeth, this is for you. We're collecting our room data at room temperature. We have to be very, very aware of radiation damage. Are the changes that we're seeing functional, or are they just a result of the changes that are being caused due to radiation damage? How do we initiate the reaction? So most biology isn't photoactivated. We can create photo cages to help us initiate reactions that way, but initiating the reactions is often a limitation. And also the exposure time. How long do we have to collect data in order to get a reasonable signal to noise in our diffraction images? And that in turn limits the time resolution of the experiments based on the source that we're using. So our time resolution in these experiments is determined by our initiation, the X-ray source that we're using, the diffraction strengths of our crystal, and occasionally the detector that we're using. So we can do these experiments now on irreversible reactions. We don't have to wait for our crystal to relax back to its starting state, reorient it, and then collect more data. We can collect single images from many, many crystals and bunch them together to get our data sets. The way that I do these experiments usually is I mount my crystals in solid supports, the position controlled with fast translation stages. We can very, very quickly scan through collecting images from each crystal on these solid supports. And as you can see over here, we can have a laser aligned at the sample position as well as the X-rays, so we can initiate our reaction inside the crystal and collect our data after different time delays. We need thousands of microcrystals. That's not always an easy task, but these experiments tend to work quite well. And as Arwen very nicely introduced, the timescales that we can watch cover the whole gamut of biologically relevant timescales, from our very, very fastest photochemical reactions that might occur on femtoseconds, all the way down to our slowest enzymatic step, seconds. And the way that we do these experiments, the places that we collect this data, so if we want to get to the highest time resolutions, we need to go to an XFEL. But if we want to do slower reactions, or if we want to do a simpler experiment, we might choose to use synchrotron radiation. So the work that I do is to develop methods to try and increase the time resolution that we can achieve using a normal monochromatic synchrotron beamline. We can do that by bunching together time points, but I don't want to talk about this method today. What I want to talk about is something that Helen mentioned, and it's, I think, particularly pertinent to when we're developing methods. We collect a lot of data and we often don't publish it because we're developing a method. So in order to develop new methods, what we need to do is have a really well-characterized system that we can then go back and test our new method on and compare. Are the changes we're seeing relevant? So when I started doing these methods developments, I was trying to look at photo-caged ligands inside enzymes, and it turns out that's quite challenging. So photo-caged ligands don't always play nice, they don't always bind well, the occupancy is often low, and we can struggle to really understand what we're seeing in our data from a novel technique. So to develop new methods, I wanted to find a protein that was amenable to photoactivation and on appropriate timescales for the methods that I was developing. So it's important to note that all proteins really are photosensitive, but is that photosensitivity really related to biology? So that's why I was looking for a new protein that was something that was well characterized, quite simple, that I could then look at and develop new methods. And so my boss at the time, Arwen, said, have you heard of crystallins? And I was sold at the word crystallin as a crystallographer. It seemed quite an attractive protein to look at. This really actually quite interesting. So this protein is found in the eye lens. So obviously its interaction with light and with UV is quite important. And it's surprisingly stable. So in the center of your eye lens, the proteins you were born with are the proteins you die with. These proteins have to remain soluble and folded throughout our entire lifetime. And with daily exposure to UV radiation, that's actually quite impressive. So what we were wanting to do is, can we understand why they're so stable, but also to look at what happens when that stability fails? Because at some point in our lives, these proteins start to unfold, they stick together, they aggregate, and they form cataract. So cataract is actually the leading cause of blindness worldwide. 1% of adults in the world suffer with cataracts. Here in the UK, in places where we have national health services, it's okay because we can get an operation to replace our lens. But there is actually no pharmacological intervention for cataracts at the moment. It's quite a controversial area of research. But what we wanted to do was to understand how this protein is stable in the face of daily UV radiation, and also what happens when that stability fails. So there's an important structural feature that we think is related to the UV stability. In each, the N and the C terminal domains of the protein, there are 2 tryptophan residues, and it's thought that these act as a sink for the UV energy that's being absorbed by the various aromatic amino acids. They can transfer their energy to the tryptophans, and then it can be dispersed through the protein. But UV is known to initiate aggregation in vivo, and it's thought that over time, very slowly, what happens is tryptophans become oxidized and they're converted finally to kynurenine. Kynurenine is more hydrophilic than tryptophan, and so this disrupts the structure, causes unfolding and aggregation. So I thought, great, I'm going to do a time-resolved crystallographic experiment. I'm going to blast my crystals with UV, and I'm going to see this conversion from tryptophan to kynurenine. Another really useful feature for somebody doing serial crystallography is this point mutation that was found in a 5-year-old patient who actually had crystals inside their eye lens. So the structure of the R36S, a surface arginine converted to a serine, was solved by extracting crystals from the patient and putting them into the X-ray beam. So quite useful for me to have a protein that crystallizes very readily, not as fortunate for the patient who had cataracts. But here you can see there we have batch crystallization. It works very well. All we have to do is have our protein in buffer with a bit of reducing agent, centrifuge them, and out crash loads and loads of microcrystals ready for me to do my experiment. So we did the time result experiment, and what we saw was nothing. So the protein is very stable to UV, and it's quite difficult to catch that damage process happening. And when we overlaid our structures, we didn't see much change before and after UV radiation. So I was thinking, how can I be a productive structural biologist? Well, I just have to collect more data and collect more data and collect more data. So at this point, I've got about 400,000 diffraction images, approximately 150,000 indexable images. My data pile is quite manageable, but I've not really got any results that I would be interested in processing, even though the methods development in order to get to this point has been quite useful. So then, okay, I want to collect more data, and my colleagues over at T-Rex very kindly said, if you want to send us some crystals, we can do a different experiment. So we did a different experiment here using low levels of continuous UV illumination. See if we could drive that damage process to happen more gently over a longer period of time. And again, what we saw in our dark structure, tryptophan, and in our UV exposed structure, tryptophan again. But here, my crystals had kind of got a bit old in the time between me growing my crystals, doing one experiment, and then sending them over to T-Rex. And upon aging, we saw something happening around this surface cysteine residue that appeared to be UV reversible. So now there's something interesting that we can start to look at, some process that maybe would be amenable to looking at using different time-resolved methods. And what we realized we could see is that we have this surface cysteine over time due to oxidative reactions. The reducing agent in our crystallization solution, DTT, actually sticks to the surface cysteine. This is not particularly something we would expect. So the oxidized form of DTT, the cyclic form, is quite stable, but that's what we saw. And then after UV initiation, the cysteine is converted into sulfenic acid. And now my data pile is growing. I've got some interesting data, but does it really mean anything? Well, it kind of relates to the natural situation. In our eye lens, we have high concentrations of a reducing agent called glutathione, not quite the same as the one I'm using, but it works by being able to reduce these cysteines, and they know that S-glutathione does bind to the protein over time, and that S-glutathione-ylated proteins have been found in aged lenses, and they know that a reduction in the glutathione concentration is found in cataract. So we've got these cysteine adducts. We think they're quite interesting, but is there anything else happening to the rest of the protein? Because it's quite important that the protein remains in its structural form in order for the chaperone protein to be able to bind to it and do its job, help it to remain stable. But so I was thinking, well, it doesn't look like there are any significant changes in the structure. And as I was discussing this with my good friend, Helen, she questioned me what I meant by significant. And what I meant was really there are no large scale structural changes that we can see by overlaying the two structures. And difference density is quite clear around this adduct, but everywhere else in the structure, it doesn't really tell us much. So what do I do now? Do I do more experiments, or do I actually start to try thinking about looking at my data in different ways? And thankfully, Helen convinced me it was probably worth looking at my data in different ways. So using her program ROPE, what we decided to do was a batch analysis to try and think about this idea of significant changes in structure. So we took advantage of the oversampling that's inherent in serial crystallographic experiments. We collect lots and lots of diffraction images. And what we could do is, by batching these together across the different chips of data that we had, we could then make repeats and boost the statistical power of our analysis. So here we can see now in this rope space that we have these clusters. And the clusters along two principal components actually relate to the different forms of our protein structure. So we have our fresh protein structure where there is no adduct. We have our dark aged protein structure where the adduct is formed. And then we have our light aged structure where the adduct has been removed, but it's not quite gone back to our starting state. And we have two chains in our asymmetric unit, so we could compare them both. And we saw these changes across both. So now we think, okay, maybe these changes are significant. The changes that we're seeing in the structure aren't just a random sampling of conformations over time, but they're actually related to the state of the crystals of the proteins inside. And so using our batch analysis, we looked at the torsion angle changes between, here you can see the differences between our fresh and aged structure without UV. And we can see in certain places there are quite large deviations in the torsion angles in our structures. And there are some important regions where we can see movement in loops. And then by doing a linear regression of our atom positions, associated with the principal component, what we could see is that upon aging, we get a contraction of the protein. And so this is quite interesting. We're starting to see dynamics are associated with this process. And so what we did then was we plotted the interatomic distances for our different states of proteins. And this is again the fresh versus the aged states. And what we saw is in our two chains, so in chain A, we saw a slight expansion in the N-terminal domain and a contraction in the C-terminal domain. And then in chain X, we saw a contraction in two of them. We're not completely sure yet why there's a difference between the two chains, but obviously the answer always comes back to crystal contacts. And then we did the same again after exposure to UV, and we saw the inverse happening. So we're seeing a return here. We're seeing a return from the expanded to the contracted states, and that's important then for chaperone recognition in our protein. And here we can see that when we generate a path that links the dark and the UV illuminated structure to look at the dynamics accompanying the UV perturbation, we can see that we have these sort of inverse dynamics associated with that process that counteract the process of aging. So this is quite interesting. This is a small change that we saw in our protein structure, and actually how something that's happening on the surface of the protein affects the global structure and the dynamics that are associated with that using time-resolved crystallography. So now I'm convinced that there is something interesting happening. I'm doing lots of time-resolved experiments. I'm doing lots of spectroscopy. My data mountain is growing uncontrollable once more. But hopefully by using these different tools and applying them to the problem, we are able to start making use of all of this characterization data that we collect whilst we're developing new methods. So in conclusion, I'm an experimentalist. Experiments are really, really fun, but data processing can be fun too if you have the right tools to see what's happening in your structures. Time-resolved experiments really take careful planning, characterization, and a lot of patience. It's often a good idea to keep going, but sometimes it's worth sitting back and really interrogating your data and thinking about what it's showing you. And collecting more data won't always help to illuminate the problem. Sometimes we do have to go back through and have a good look at our old data and think about the best ways to analyze your data. So for time-resolved experiments, a difference map, an overlay isn't always going to tell you the full story, as has been repeated multiple times during this meeting. It's important to have a look at different ways that you can analyze your data. So I'd like to say a big thank you to my PhD student, Jake, and his other supervisor, Yvonne, who are biochemistry experts, have really helped push this forward with starting to think about different point mutations we can do to interrogate the mechanisms that we're looking at. Owen Pearson for introducing me to the idea of time-resolved crystallography and being a massive help. Helen as well for the programs that she's developed that have made it possible for me to start to really interrogate this data. And Godfrey Bedard, my photochemistry expertise that's always on hand to give me some advice, and also all the people who've given me moral support along the way, the Beamline scientists, and Wellcome Trust and Academy of Medical Sciences for funding. And finally, if you know of any good master's students, please point them in my direction, because I've got a PhD project for doing different time-resolved methods development. And thank you all for listening.
Thank you, Briony, for this fascinating talk. I can see that there are already questions. Good luck, because we have time for these.
Thanks. Ata Acero from City University of New York. So I had a question about the age of the crystals, and also how are you storing the crystals if they're like, because the light, was it in light? Did you put them in the dark?
They were just in a box, honestly. The crystals at the point of the adduct formation, it tends to take about 9 months. Obviously, this is a lot faster than is happening in the islands. The islands is far more complicated than just my protein in a crystal, but we think it's probably similar to what happens over a much longer time scale.
And then one little add-on question. Do you think you're going to go back and check after like a year or two years?
Oh, yeah.
Yeah.
Yeah, I have so many crystals that it's just worth keep going.
Thank you. I think more questions on the shop floor.
Hi, Mike Cuff from Diamond. I thought it was really interesting that your crystals seem to cluster either into fresh or aged.
Yes.
Is there any kind of mixture or intermediate states on the way?
Yes, there are indeed. So we've been doing some time-resolved experiments on faster processes, but I think what happens is that it's quite difficult to know exactly when the crystals are going to be in that intermediate state between fresh and aged, because it takes quite a long time. So I think, yeah, just keep going and trying to catch it along different parts of the reaction. What's really interesting to me though now is trying to see the difference between light and dark and trying to actually capture the mechanism of the photochemistry that's happening as well.
Great. I think we have a couple of questions on Slack.
There's another one in the room. You can answer the Slack ones later if you really want to.
So Elke Zitter from the IBS. Did you only calculate the standard 2FOFC and FOFC maps, or did you try other approaches as well as FOFO maps and so on?
No, at the moment I've just done the FOFO, but I'm definitely going to try doing some extrapolation.
Because it might be that there is a small signal which you can extract with those kind of maps.
Yeah, I'm desperate to go home and start trying that now.
Any more in the room?
That was really fantastic. Thank you. I wondered about the chaperone protein that you mentioned, and where that actually, where the interface with them, what you showed us is, if that's known, and whether that could affect the movements that you're seeing.
Yeah, the chaperone protein is really interesting. So it forms oligomers, but it deliberately appears to form oligomers of very varying sizes to prevent crystallization in the lens, because it's at such high concentrations. So we don't have a good idea of the structure of the chaperone protein, but we think that it's really important for the N-terminal domain to be in its native conformation for it to be able to bind. And again, there is some discussion about whether it sequesters unfolded proteins and then just prevents them from sticking together, or if it helps to refold them. And so these are experiments that we want to do, and hopefully then this is sort of Jake's next step.
Matt McLeod asks, have you grown crystals in glutathione to see if there is a difference between DTT and glutathione?
Yeah, we've been trying to grow crystals in glutathione. I think we've just about now managed to get a structure of the glutathione bound. The process of glutathionylation seems to be much, much slower than the DTT addition. It's bigger. I think the actual structural changes that occur are actually much larger, and it might have more of an impact on the domains and the changes from the native step. But yeah, that is definitely on the way.
And Miranda Lynch asks, in the batch analysis under the rope analysis, the pronounced spread in the two fresh structures is noticeable in the clustering. Does that imply that those structures themselves are more variable, or that the motions are more pronounced?
I think it just shows that we might not have enough data from the fresh structures to be able to see the clustering very well. But there may be more variation in that structure than the other two.
And Mike S asks, can you accelerate the chemical changes using environmental shifts such as increased O2 or temperature?
Yeah, absolutely. And changing the pH is one of the easiest ways that we could do that.
Any more questions? I have one.
Okay.
Is there any way, and if so, how, to deconvolute the differences between individual molecules in the asymmetric unit and the real shift in structures due to real proper change?
Yeah, we hope so. I think this is where you have to start looking at new other methods and bringing in sort of complementary data to be able to do that. You really want to be able to see changes in solution as well, and then high concentrations of solution without aggregation. So it becomes quite complex. But I think something that I would like to move towards is sort of direct imaging. So in sort of cellular environment. So yeah.
Thank you. Any more questions? If not, let's thank Briony once more.